Welcome to our digital event this evening. For those who have been here before, you'll know that we like to start off our digital events with a whakatauki, and this one is what sits above our head of research's desk in her office, and it goes, Ihara, takutoa, iti toa, takitahi, engari, he toa, takitini. And that translates to my strength is not as an individual, but as a collective, which we find to be very appropriate in the research world. So as you can see, your camera and your microphone have automatically been turned off, and you won't be able to turn these back on for the duration of the event. That does mean that we cannot see or hear you. But if you do want to communicate with myself during the event, you can use the chat or Q&A functions. You'll find these at the bottom of your screen, just a few little icons down there. You can use the chat icon. It'll pop up a window. You can use this to talk to me if you have any technical issues. No other attendees will be able to see what you put in the chat box. It's set so that you're only speaking to myself as the host and the panelists. And as I mentioned, you can use it for technical support questions. I'll do my best to help you. If you have any questions about today's topic, at the end of the session, there will be a Q&A, and you can use the Q&A function for this. So that's at the bottom of the screen as well. Just again, click on the little icon. It'll bring up a pop-up box, and the questions will be visible by all the attendees, and we will be able to see who has submitted the question. However, if you wish, you can tick the little tick box that says to send anonymously, and then it will come through anonymously. Once your question is submitted, we will opt to answer the question live so everybody can hear, or we might type a response to you, so just keep an eye on that. And also keep in mind that we have a limited time frame tonight, so we might not be able to answer all of your questions, but we will do our best. And additionally, please keep all questions related to the topic presented. Our speaker, Sophie, will not be able to answer any personal medical related questions that you might have. And at the end of the session, the Zoom will end for everyone. Those who have been here before know it can be a little bit abrupt, so apologies in advance. And there will be a recording available on our website later this week. So if you missed it or you want to watch again, please feel free to check that out. Okay. Now, a little bit about our speaker this evening. For those who received our Headlines magazine, which came out earlier this month, you'll know a bit about Dr. Farrow and her research. But Dr. Sophie Farrow is with us tonight, a postdoc fellow at Liggins Institute in Auckland. Her early career research was supported by a Neurological Foundation small project grant. She's now supported by the Michael J. Fox Foundation with a grant to study Parkinson's and genetics. And her work was recently published in the neurology journal Brain, which is very exciting. So we are very glad to have you here with us today, Sophie. And I will hand on over to you and just let me know if you need anything.
Great, thanks, Juliet. Okay, I'll just share my PowerPoint now. Okay, can you see the right screen?
We can see your presenter view.
Okay, let's swap those. Better?
Perfect.
Cool. Okay, so thank you, Juliet, for the introduction, and thank you to the Neurological Foundation for inviting me to speak today. So as Juliet said, I'm a postdoctoral fellow at the Liggins Institute, and my research really focuses around Parkinson's disease and the genetics that underlie Parkinson's. So this is sort of an overview of the talk I'll give to you today. And so I'll start firstly with the introduction and background, just very briefly. So what is Parkinson's disease? So Parkinson's disease is the fastest growing neurological disorder worldwide, and it currently affects more than 7 million individuals. And in 2018, to really reflect how quickly the disease is growing, these two experts in the field wrote a paper called The Parkinson's Pandemic, which spoke about how quickly the incidence rate is rising and predicted that by 2040, there will be almost around 13 million individuals affected. So this has just shown that the incidence is going to rise by almost double in a matter of about 20 years. And that's very reflective of the aging population. And in very simple terms, pathologically, Parkinson's is defined by the alpha-synuclein of that alpha-synuclein accumulation within Lewy bodies, and also the death of dopaminergic neurons. And clinically, it's defined by 4 cardinal motor symptoms. So the bradykinesia, or the slowness of movement, muscular rigidity, resting tremor, and postural instability. But it's also defined by these non-motor symptoms, such as constipation and fatigue. And across different individuals, we see a huge amount of heterogeneity, particularly in these non-motor symptoms. And then finally, the most common form of Parkinson's is this idiopathic or sporadic form of Parkinson's disease. And the rare familial form, which we often consider to be inherited across generations, is only seen within about 5 to 10% of all Parkinson's cases. So I'll focus a little bit more on this sporadic versus familial form of Parkinson's now from a genetic standpoint. So when we consider Parkinson's from this genetic aspect, there are the 2 main forms, as I said, the sporadic and the familial. And the familial form is particularly rare, but it's also fairly well characterized, and that we can typically attribute the cases to monogenic causes, such as mutations within the alpha-synuclein or the LRRK2 gene. And then sporadic or idiopathic essentially implies that the disease is of unknown etiology, and it typically affects around 1 in 100 individuals over the age of 60. Now, despite being described as sporadic, there are several risk factors that have been identified at increasing your risk of Parkinson's. And these include exposure to specific toxins such as rotenone, and also things like head trauma, and that's being particularly explored in New Zealand at the moment. And there's also some genetic risk factors which have been associated with the risk of sporadic Parkinson's disease. And this is really where my research focus lies. So I'll be talking a lot about this as we go on through this talk, as well as the GBA gene, which tends to link across the 2 different forms of the disease. So firstly, I just wanted to talk about a few different hypotheses or ideas that have sort of driven the work that we've done so far and the work that we plan to do. So this is really talking about different genetic variants and how they function within larger regulatory networks rather than just acting as a sole individual, and really understanding how an individual variant can affect a gene within a pathway, such as here, but then it might have a much larger knock-on effect depending on the proteins or the genes that are interacting with this one gene that's initially targeted by the variant. And then if we consider that some individuals have multiple variants that affect multiple genes within the same pathway, then understanding this can really lead to understanding as to how some pathways are so badly affected in some individuals because they've got multiple variants affecting multiple genes within the same pathway, whereas other individuals might just have 1 or 2 variants. So I'll come back to this later. And then this second hypothesis really links to the title of this talk. So talking about how the effects of these genetic variants associated with Parkinson's aren't necessarily limited to the brain or the neurons, as we might think, given that Parkinson's is a neurodegenerative disorder. And this really is in line with lots of different groups, different research groups across the world that have been trying to identify an enrichment in brain cell types for Parkinson's disease. And typically when we look at diseases such as Alzheimer's or schizophrenia, we identify this strong enrichment of genetic variants in the brain cell types, such as neurons. But when they've looked for Parkinson's disease, there's not necessarily this enrichment within the neurons. And this has led different researchers to hypothesize that it's likely that these variants are having more of a global effect across different cell types throughout the body, rather than just this focus on the brain. And this could also explain quite a lot of the non-motor symptoms that we see that aren't necessarily linked directly to the neurodegeneration. And then also I wrote in this title here, neurons as opposed to the brain, because there are different cell types within the brain, such as microglia, astrocytes, and oligodendrocytes, which have been identified as being important in Parkinson's, as well as the neurons. So looking elsewhere within the brain as well as elsewhere within the body. And then the final hypothesis is this idea of Parkinson's disease being an umbrella term, and describing it as this umbrella term for the multiple different subtypes or disease types that are present within this Parkinson's disease. So it's essentially in this mountain model is highlighting the many potential origins and factors that might contribute to the observed heterogeneity we see in Parkinson's, and the different origins that might lead to, therefore, different subtypes of Parkinson's disease. So at the moment, the vast majority of Parkinson's disease research is really focused on this endpoint of Parkinson's disease. So looking at the alpha-synuclein buildup in the Lewy bodies and the death of the dopaminergic neurons, and then the clinical symptoms that arise from this. And at the moment, when people have been trying to define subtypes of Parkinson's disease, they really focus on these clinical manifestations and trying to see if you can break individuals into different groups based on how they're presenting clinically. But by the time an individual's actually developed all these clinical symptoms, they've likely already had the disease manifesting for like 5, 10, 20 years. And so it's likely too late to start treating these individuals based on the subtypes. So if we can start to move towards this more bottom-up approach, by which we can subtype individuals based on their genetic risk profiles, or perhaps protein or lipid biomarkers, if we can identify some, then we can begin to develop these biological subtypes rather than clinical subtypes. And this agrees with a couple of clinical trials that are currently being conducted in America, where they're trying to base individuals, group individuals based on how they, their biomarkers in their blood are grouping as opposed to the clinical presentation. So now I'll just talk about a couple of concepts that really underpin the research that I've been doing and the research within our group. So firstly, very simple, when I talk about variants, I'm typically referring to these single base changes within our DNA. So here we have a T allele and it's changing to a C. And this is a single base change or a single nucleotide polymorphism. And we often call these SNPs. So you'll probably hear me talking about them as SNPs throughout, but they're just variants within our DNA. And then the concept of the non-coding genome. So within our whole genome, actually only about 1% of our genetic code is coding for proteins, and the rest of it is non-coding. And this is essentially important to understand because if a SNP or a variant lies within this non-coding region of the genome, it's much harder to assign function to what the variant is doing and the downstream impact. And then the next important aspect that's key to understand is the three-dimensional structure of the genome. So often when we think about the chromosomes inside our nucleus, we think about them being organized in this nice structured layout, similar to if we think about the Earth being flat. However, in reality, our chromosomes are organized into a three-dimensional hairball-like structure inside the nucleus. If you think about something like an elastic band ball, or yeah, as I say, a hairball. And this structure actually enables interactions to occur between chromosomes, and knowledge of these interactions is instrumental for the association of genetic variants or SNPs with their respective gene targets. So as I just mentioned, the physical interactions between the different chromosomes or different parts of a single chromosome are instrumental for gene regulation. And so if the variant in the gene are near to each other on the same chromosome, we call this cisregulation. So here you can see this is the variant and this is the gene being affected, and they're very nearby. And this is the hypothesis that's sort of been used for many years, by which you assume that the SNP or the variant is affecting the gene that it's directly next to on a chromosome. But using the knowledge of the three-dimensional genome, we can also have interactions that happen still on the same chromosome, but much further away, so more than 1 megabase away. So you have the SNP, and then you have a big loop occurring, and then it comes into contact with the gene much further along. And then perhaps the most novel set of interactions that can be identified using this physical interaction is the a SNP being on, say, chromosome 1, coming into contact with a gene on chromosome 6, and therefore affecting the gene expression due to this physical interaction occurring within the hairball. So hopefully they were relatively clear. So now I'll go on to talking about how we use those concepts and identify target genes for risk loci through integration of publicly available big data sets. So this is quite computationally focused, and then I'll move more on to the lab work that I've done towards the end of the talk. So coming back to this slide that I showed just a minute ago. So as I mentioned, if a SNP falls within the non-coding region of the genome, it's particularly hard to assign function to. And given that 99% of the genome is non-coding, that means majority of the SNPs lie within these regions. So our lab, along with many others, are trying to tackle the challenge of assigning function to SNPs. And so we use an algorithm that was developed by our lab, and this is known as Code3D. And essentially it takes chromatin conformation data sets, which are the data sets which actually have captured this hairball-like structure of the chromosomes within the nucleus of different cell types. And these are all publicly available. And once we can take these data sets and actually identify the physical connections between the SNPs and the genes. And from this, we can identify SNP gene pairs. And then we can take the pairs and see in expression data sets if the SNP and the gene, if the SNP is actually having an effect on the expression of that gene in any particular tissue that we're interested in. And so this is using publicly available data sets, and they have very rich data for a whole number of different tissues. So the first application we worked on using this algorithm and principle is the GBA locus in Parkinson's disease. And the reason we focused on the GBA locus is because it's the most common genetic risk factor in Parkinson's disease. The mutations are typically found within between 2 and 9% of all Parkinson's cases. But the issue with the mutations in the GBA gene is that even if an individual is carrying a GBA mutation, only around 21 to 29% of all patients will actually, of all individuals will actually go on to develop Parkinson's disease. And this is a phenomenon known as incomplete penetrance. And it's also a pleomorphic gene, which indicates it's involved in both the familial and the sporadic forms of Parkinson's disease. So this is a map of the GBA gene and the loci outside of it. And we mapped across the loci all the common SNPs that are occurring, and we identified 128 different SNPs that are occurring across this GBA locus, and these were considered common SNPs that occur in more than 1% of the population. And using the algorithm that I spoke about earlier, we identified 73 out of these 128 SNPs that were having a regulatory effect on at least one gene and at least one tissue. So this map looks a bit mad, but essentially it's showing all the different, all the different genes that are affected by these variants and all the different tissues across the body. And we identified a number of different variants that were associated with the altered expression of GBA, which may in part explain some of the incomplete penetrance associated with the GBA missense mutations in Parkinson's disease. So, for example, if you have a missense mutation in the GBA gene, but then you also have a couple of these other variants affecting the GBA gene, then it could either exacerbate the effects of the original mutation so that you get Parkinson's disease, or weaken the effects so that you are protected. So also within this piece of research, we collaborated with some colleagues down at the Uni of Otago, but in the Christchurch branch, who had genotype 229 Parkinson's patients. And they'd really focused on the sequencing of this particular GBA loci. And from this, we identified 3 of the non-coding SNPs that were associated with the expression of GBA and other genes across different tissues. And these SNPs form a haplotype, i.e. they're inherited together, that's significantly associated with the age of symptom onset and age of diagnosis of Parkinson's disease. So as you can see here, depending on what genotype you have present or what variants you've got, you might have a diagnosis age of 67 or 61. So there's quite a big contrast between just having these three variants. So now moving on to a more broad approach to understanding Parkinson's disease. So genome-wide association studies are conducted for all diseases, pretty much, and they essentially identify variants that occur more frequently in individuals with the disease versus a healthy control. So the most recent genome-wide association study, or GWAS, for Parkinson's disease was conducted in 2019 by a group led by Mike Nowes in America. And they compared the genomes of 37,000 Parkinson's cases versus 1.4 million controls. And they identified 90 SNPs across 78 risk loci that were associated with Parkinson's disease. And so I've included this figure here, which came from a recent review by a couple of guys at Stanford University. And it's essentially highlighting the pipeline to move from genome-wide association study identified SNPs right the way through to how we actually, right the way through to understanding how they actually affect the trait or the disease that we're interested in. And so the work that I'm talking about at the start is really looking at this computational prediction and computational prediction and what target genes we think are happening. And then as I move through my talk, I'll talk more about the screening assays and then how we can actually use gene editing to predict more precisely the causal genes and the function of these variants. So here, this is just the image of the Parkinson's GWAS that was conducted that I spoke about just a moment ago. And these are the 90 different, or the 78 different risk loci and the 90 different SNPs across all 22 chromosomes. So I took these 90 risk loci or SNPs and explored the regulatory impact of them on gene expression using the concept of the 3D genome to aid in identifying the target genes. And in doing so, I identified 518 genes that were targeted by these 90 SNPs. And this really highlights that a single risk loci can contribute to the regulation of more than one gene. And also a single gene's expression could be controlled by several different variants. And then I used pathway enrichment tools to understand which biological pathways are likely to be affected as a result of the alterations in gene expression. So this is just highlighting what I spoke about just then. So the 90 SNPs, we've got the 90 SNPs and then 76 of these 90 are regulating genes. And in total, we have 518 genes. And I think quite interestingly here, we have 178 of the 518 genes that are being regulated through these trans mechanisms that I mentioned earlier. So the long distance mechanisms occurring between different chromosomes. And so this set of genes that were identified through these mechanisms are fairly novel to the work that we've done. We then took the 518 genes and looked at the protein products of these genes to see how they interact with each other based on publicly available experimental data sets. And from these interaction networks, we used a clustering algorithm, which allows us to identify the genes and their protein products which are interacting with each other, and if certain clusters of the genes and protein products are forming based on these interaction networks. And so using this approach, we identified 9 significantly interconnected clusters, and these were each enriched for specific biological pathways. And I guess what we were trying to really find out through this is using the concept that different Parkinson's associated variants each contribute to the regulation of the genes within each cluster. And so if an individual has a specific combination of risk SNPs, then this may lead to detrimental effects in some of the highlighted clusters, which in turn would affect the enriched biological pathway to a lesser or greater extent. And this comes back to the first hypothesis that I spoke about, that we need to look at the effects of the variants within a much larger network setting rather than just seeing the direct target gene of them. So some of the clusters that we identified were enriched for relevant pathways to Parkinson's disease, such as synaptic vesicle recycling and immunological surveillance. And then different clusters were enriched for microtubule polymerization, which is a fairly novel pathway for Parkinson's disease, but has recently, in the last year or so, gained some traction. And we can also see here these orangey red nodules are the genes and their protein products that were regulated through these long distance trans mechanisms. And as you can see, they're fairly central to some of the clusters forming. So it's really important to consider both the nearest genes and also the long distance interactions occurring for identifying these networks. So now I'll move on to some more of the experimental work that I've done. And this section in particular was funded by the Neurological Foundation Small Project Grant. So thank you for that. It's yielded some quite interesting results. So one of the problems with genome-wide association studies is linkage disequilibrium, or LD. So I'll explain this concept very quickly. So LD essentially refers to variants or genes that are inherited together more often than expected by chance. And this results in a non-random association of variants. So a different way to think of this is that if we think of the variants as puzzle pieces, normally when you mix up puzzle pieces, they can form different pictures. But if two or multiple different specific pieces are always stuck together, then they will always be found together and always form the same picture repeatedly. So in a similar way, if we have two or more variants in linkage disequilibrium, then they tend to be inherited together more frequently, and their specific combinations become more common in a population. So this is essentially what this is showing here. So you could have one variant that's in linkage disequilibrium with a whole number of different variants, and this forms a larger linkage block. And this is problematic for genome-wide association studies, because typically when they report results, such as the 90 risk SNPs that were identified for Parkinson's, these are actually 90 tag SNPs that are part of much larger linkage blocks. So although the reported tag SNP is usually the most statistically significant association at that particular loci with Parkinson's disease, it doesn't necessarily mean it's the causal SNP within that block. So this highlights this here, how you've got lots of different SNPs within this genomic loci. And this one here would be the one that's reported as the tag SNP because it's the most significant or has the strongest signal. But in reality, the causal variant might not be this one. So one way to try and tease apart these linkage blocks is a method known as massively parallel reporter assay, or MPRA. So MPRAs are essentially trying to work out which variants are the important variants on a quest to identify how common variation affects a disease phenotypes. So reporter assays typically in the past have explored risk variants, and they've had to do this in a one-by-one manner. So you take one risk variant and look at it individually. But this massively parallel reporter assay allows us to look at hundreds of thousands of variants all at once. And then we can systematically evaluate the individual variants and identify which ones are likely causal and which ones are likely passengers along for the ride during the selection process. And I think it's important to think about the common variation or the genetic variants as tuners of gene expression, like a dimmer switch. They don't tend to turn gene expression completely on or off. So, so massively parallel reporter assays are essentially a high throughput tool to simultaneously characterize the regulatory activity of thousands of different variants. And this takes advantage of the well-established reporter gene assay, by which we synthesize a library of barcode tagged putative regulatory sequences which contain the SNPs of interest. And then these are directionally cloned into reporter plasmids. And then these can be transfected into the cell type of interest. And so we use TEC293 cells, which aren't necessarily the best choice for Parkinson's disease, given that they're embryonic kidney cells. But for the massively parallel reporter assay, you really need a high transfection efficiency. So they are a great starting point and have previously been shown to actually mirror neurons fairly well when compared to other different cells that you might use for an MPRA. And then high throughput sequencing of the transcribed barcodes can then be used to determine whether the variants are acting as enhancers. And using lots of tag barcodes for each variant in the library allows us to decrease the noise and get an accurate readout of the activity of each variant, because you can average across the different barcodes that are all linked to the same variant. Okay. So within my MPRA library, I focused on the 90 Parkinson's loci and identified all the SNPs that were in linkage with the tag SNPs. And so this is just highlighting all the different loci across the genome. And for some, you can see there were lots of SNPs at a particular loci. So for this one here on chromosome 5, we have around 300. And then for others, there were much fewer linked SNPs. So I took this library of around 5,000 candidate variants and followed the process. And then on the left here highlights the pipeline from the original library with 5,000 pairs of variants right the way through to the final identified elements. And throughout this pipeline, we used very stringent cutoffs, especially for the alignment of barcodes to their respective elements, which has resulted in the identification of a fairly small number of regulatory elements. But this does mean that we can be very confident in the enhancers that we have identified. And just a note here, when I'm talking about these enhancers, it's again the, if we go back to the dimmer switch analogy of turning the gene expression up or down, depending on which allele is present. So, this just highlights here all the enhancers that are acting in an allele-specific manner. So if we identified the allele-specific enhancers for 27 loci at an FDR or significance cutoff of 0.05. So all the little red dots are significant enhancers at the loci. So I'll show you one specific example here. So this, if you remember back to the linkage block, this is a linkage block associated with this SNP here, RS2904880. And this SNP falls within the coding region of the CD19 gene. And for this SNP, which is the tag SNP of one of the 90 GWAS loci, we identified no enhancer activity. So there was no differential allele activity at all. But this SNP over here, RS11646653, which is in very strong linkage, so inherited together with this tag SNP, we identified very strong allele-specific activity, enhancer activity. And so we can see here, this is the difference between the genotypes, and this is showing the significance value for this, so it's acting in an extremely significant way. And this here highlights one of the potential mechanisms by which this might be acting. So we can see here, this is in the reference genome. So we have a C allele, and then in individuals with the SNP, they might have the T allele at this particular position. And this changing of allele is disrupting this binding site for the ARNT transcription factor binding site, and therefore weakening the enhancer activity of this particular loci. And some potential gene targets for this loci include SH2B1 and NFATC2IP, which are involved in vesicle trafficking and also positive regulation of transcription by RNA polymerase. So just some just highlighting how it's important to look beyond these tag steps for identifying the actual causal variant and then downstream gene targets of those. So the work funded by a neurological foundation that I've just been speaking about really allowed us to apply for a larger grant from the Michael J. Fox Foundation, really looking at understanding how, once we've identified these enhancer variants through the use of the MPRA, how they're actually having those impacts on what genes they're affecting within a relevant cell model for Parkinson's disease. So to do this, we use induced pluripotent stem cells, or iPSCs, and we're using a line known as COLF 2.1J, which is a cell line that has been derived by a group in America to be the ideal cell type, cell line for introducing these different variants, because it has no known Mendelian mutations. And so a lot of groups are using this particular cell line with the idea that we can create a catalog of different variants within this particular cell line. And so we take our induced pluripotent stem cells, and then because we're interested in just these single base changes, we can use gene editing or CRISPR-Cas9 to introduce these variants. So we're literally just changing that single base within the genome. So we have isogenic pairs that differ only at that single base. And then we can take these edited cell lines and differentiate them initially to neurons, and then also to different cell types such as astrocytes or microglia, and then measure gene expression in these cell types using RNA-seq to understand the impact of these edits or the introduced variants on gene expression, to see which genes are actually affecting and if the affected genes fall within a network. And then also, given that variants don't necessarily act independently, we hope to introduce multiple variants into the same cell line to see if there's any additive or interaction effects occurring between these. So I wanted to sort of just summarize this talk by talking about what the end goal is. So I'll come back to this mountain model talking about the subtypes of Parkinson's disease. So we have this umbrella type of Parkinson's disease, and this is really talking about the heterogeneity of individuals, and that no individual with Parkinson's disease has the same trajectory or the same set of symptoms, and that we want to understand this from a biological standpoint as opposed to clinical. So the idea is that we can take these panel of genetic risk loci that have been associated with Parkinson's disease, and then one individual might have one set of genetic risk loci associated with Parkinson's, whereas this individual would have a different set, and so on, so on. And then based on the genetic variants or risk profiles of these individuals, we can identify the target genes and therefore the pathways associated with the variants an individual has, and based on the biological pathway affected, which is likely the causal pathway for their Parkinson's disease, you can base treatment on this biological pathway that's being affected. And so moving towards this precision medicine approach for Parkinson's disease, and moving away from the one-size-fits-all approach that is typically used. And so, although we say everyone has Parkinson's disease, we're moving towards having everyone being in a different subtype within this based on their biological definitions. So to summarize, this image here is done by a guy known as Johnny Aitchison, who is a doctor that's been researching and helping individuals with Parkinson's for years, but has recently been diagnosed with Parkinson's, and he's developed this set of images just to really highlight the complexity of Parkinson's disease beyond the 4 cardinal symptoms that are typically used to define Parkinson's. Sorry, little side note. So to summarize, the key aspect underlying a lot of our work is that considering the 3-dimensional structure of the genome really allows us to identify this novel set of target genes across many different tissue types. And using this, we managed to identify a group of 3 variants within the GBA loci that are associated with the age of symptom onset and diagnosis age. And we can use similar approaches to what I spoke about for the GBA locus, but apply it to different gene sets to see if other variants within different known risk genes for Parkinson's are actually associated with different clinical outcomes. And then the MPRA analysis enables us to identify potential causative variants within these really complex GWAS loci. And also an interesting point that I haven't mentioned yet is that the identified enhancer variants typically weren't the original tag variant identified by the genome-wide association studies. So it's really important to dig down beyond what we see at the face value. And finally, we can use these induced pluripotent stem cells and gene editing to model the effects of variants in neurons and other cell types to really understand the functional mechanisms by which these are working. So finally, I'd like to thank the group that have really been involved with this work, and Justin O'Sullivan and Anthony Cooper, who have really led this, and the wider team here at the Liggins Institute, and also to all the funders, of course, Neurological Foundation and Michael J. Fox and the Dines family philanthropic funding. And thank you all for listening. Looking forward to any questions. And if you're interested in reading more about the research that I've done, it's published in these 3 papers here mainly. But yeah, thank you so much.
Thank you, Sophie. That was so interesting. I feel like I had a bit of a crash course in genetics, which was really exciting to hear about the work that you guys are doing. Some really key things jumped out at me, like working with the teams in Christchurch from the Otago Uni, just really highlighting how much the kind of collaborative approach to research is so important. You know, you're pulling information from here and down there, and obviously we've got the Michael J. Fox Foundation involved. So it's really remarkable work that you're doing. So for everyone at home, we are now open for questions. So I see there's a few there, so I will just jump in and have a look. Okay. So the first one, I'm hoping you're familiar with the term junk DNA. Is it there to appropriately place the active genes in 3D space? Can you give us any insight into that?
That's a good question. So junk DNA was very much a term that a lot of people still talk about, but essentially the junk DNA is referring to this 99% of the genome which is non-coding. And so yes, the non-coding regions of the genome are critical and are organized in a very specific manner. It's not random, to actually orchestrate how the genes are regulated and how they're, depending on how they're located within this 3D space is critical for how they're, whether the gene's up or down regulated, and it's also tissue specific, which is why we look across all sorts of different tissues.
Thank you. Okay, a question about the how. How do you change the specific bases in a gene? Is it a lab process done by hand or is it done by a machine?
So it is a lab process, sort of done by hand. So you use the CRISPR-Cas9 protein, which is essentially a method to sort of, in a simple way, cut and paste DNA. So you have guide RNAs which direct this protein to a particular part of DNA that you want to edit, and then it essentially cuts open and replaces it with a template strand that has the one edit that you want to introduce. So it's all designed by hand, but to actually introduce this into a cell type, you use a cell line. We use a machine known as a nucleofector. So essentially done by hand, but unfortunately, it takes a long time. But there are more high throughput methods that are coming now, so known as CRISPR screens, but they're kind of high throughput for introducing hundreds of different variants. So if you're just interested in a couple, then you wouldn't want to use one of those screens.
I see, more precise.
Yeah.
Okay. Are there any precision medicines in the pipeline that might help those living with Parkinson's in the near future? Do you know much about that?
For some people with specific mutations, so more on the familial side of Parkinson's disease, there have been a few trials. So one that I know a little bit about is focused on the GBA gene, and they're trying to, this is a group in the UK, they're trying to use a drug known as Ambroxol, which was already designed for a different disease, and it's been repurposed for Parkinson's disease. And it increases the activity of the GCase enzyme, which is associated with GBA. And so if you have that specific mutation, then they're trialing that drug in those individuals. And I think there are a couple of other trials that are being done like that. And then there's a couple of other trials, such as the Australian Parkinson's Mission, which are trying to take people's genetic risk profile and actually decide which drugs are likely going to work based on the genetic risk profile of an individual. So there is a movement towards that, but it's still very much in the clinical trial phase as opposed into the clinical treatment space.
Sure, and these things take time.
Yeah.
How much international collaboration has your grant from the Michael J. Fox Fund opened up, bringing that knowledge back to New Zealand? It's quite exciting.
Yeah, so we've actually been working with a group over in America, which is, I guess, where the Michael J. Fox Foundation was set up. And that's very much with the CRISPR and the stem cells side of things. So we got our cells from that particular group. And they do a lot of work around Parkinson's disease genetics, similar to what we're doing. And we also have collaborators in Germany. So it has opened up quite a lot of international doors, and hopefully, as I mentioned briefly, the cell line that we're using, they're trying to create really this repository of variants within the cell type. So as we feed back data to that group, hopefully, then we can build on this collaboration. If we find cool things, then they'll share things back with us, sort of thing. So yeah, it's quite an exciting space to be in.
Yeah, it's very exciting. And again, highlighting that collaboration, and it's just so important for everyone to share that knowledge and share everything that you learn, and so we can keep progressing. It's remarkable.
Yeah, and I think especially important in New Zealand, which is a relatively small country, actually harnessing different connections that we can get to learn different techniques.
Absolutely, yeah. So back to the sort of drug repurposing a little bit. Someone's got a question here about a specific medication, but I'm unsure that we'll be able to answer that one, depending on how your knowledge of it, if you have any. But for that person, please do get in touch with us off air, and I'll see if there's somebody that can help answer your query.
Yeah, I think I can see the question you're talking about. I don't know specifically about that drug, but we have had some ideas in the past with individuals that have been on drugs like that have to have known Parkinson's side effects. I mean, I don't think those drugs are necessarily used much anymore, but actually seeing if there's a genetic interaction with the target of those drugs, and seeing if we can understand more of the genetic basis, or if it's if there is a genetic basis underlying why those drugs are linked to Parkinson's effects. But I could if I'm happy to talk about it more after.
Cool. As I say, please get in touch with us at the foundation, and we can see if we can get more information for you. We've got someone here who very generously is offering to be a part of any research. So thank you. And if you are interested in participating in research, you can get in touch with the research institutes directly. Is that right, Sophie, with the Liggins and with Centre for Brain Research?
Yeah, I think in terms of Parkinson's disease at the Liggins, we're very much at the cellular level right now, so it wouldn't be clinical trials. But I know, the CBR might be, Centre for Brain Research might be doing more clinical trials. And I think even keeping an eye on international trials is just interesting to see what's about, and if something is close to being passed, then it might then come to New Zealand. But there is a lot of exciting things happening in the clinical trial space, so hopefully a couple of those will reach approval soon.
Yeah, fingers crossed. And unfortunately, we're a little too advanced for Sophie's work at the moment, being just simple things, simple living things. So, but thank you for your offer. And it's such an important part of research, as we know, when things get to that stage. Heading back to the gene analysis, what are the implications for people in New Zealand living with Parkinson's, and will there be better improvements in diagnosis? I suppose when you think about your mountain slide, it's that early intervention, early prevention, and targeted kind of specific.
Yeah, so thank you for that question. I guess that really underlies the whole premise of what we're trying to do. We want to improve the diagnosis of Parkinson's in New Zealand and further afield. If we can use if we can identify genetic risk factors that could lead to identification of biomarkers, or actually genetic risk profiles that will inform us about people that are more likely to get Parkinson's disease, then we can start to identify them long before the symptoms actually present, or perhaps faster diagnosis, because often individuals go to the doctors and then aren't diagnosed for a couple of years. So if we could have a test at that first stage, then we can not only diagnose earlier, but also the interventions that typically haven't worked so well would likely work much more effectively in the earlier stages of the disease.
Yes, yeah, absolutely. And so important. And wouldn't it be exciting if something like that got its start in New Zealand? Thank you, Rich, for that question. That's our CEO at the foundation. Nice to see you, Rich. Okay, how far are we from using the genetic information from using in Parkinson's disease diagnostic? Well done, Sophie.
I think in a lot of senses we are actually fairly close, not necessarily the work that we're doing, but other groups that are also exploring similar sort of things. So as I mentioned, the group in Australia, the Australia Parkinson's missions, they're very much using genetic risk profiles to inform which drugs they want to try on different individuals. So at the clinical trial phase, I think genetics are really being used to push our understanding. And also a problem that has previously been noted in clinical trials for Parkinson's disease is that no genetic information has been collected. So actually, and then they fail. So we want to understand from a genetic perspective why they failed, but if there's no genetic data, then we can't. So I think the vast majority of clinical trials being done now are collecting that genetic data, so then it'll be much easier to understand from a genetic perspective which individuals are responders versus non-responders. So I think it's already being applied in many instances.
Awesome, thank you. Yes, that's very exciting. A question about the familial genes for Parkinson's. I know we covered a lot of the sporadic genes, but can you define how you label this in a simple way, if possible?
So the familial form of Parkinson's disease is typically the earlier onset form of Parkinson's disease. So individuals that develop Parkinson's before the age of 50. And this is often also associated with a slower trajectory. So say you were to get Parkinson's in your 20s or 30s, we can use Michael J. Fox as an example. So he's had Parkinson's for over 30 years now and is still doing relatively okay. Whereas someone that gets it later in life is much more likely to have a much faster trajectory for the disease. And what was I going to say regarding this? Also, the majority of the Parkinson's that are familial, they can be attributed to, say, 1 or 2 mutations within a known Parkinson's gene, such as SNCA or LRRK2. And there is a list of around 20 genes that are really associated with this Parkinson's. Whereas the sporadic form of Parkinson's, there's a whole list of different variants rather than just 1 or 2. So yeah, it's quite defined, and often you would know if you have a more familial form of Parkinson's disease based on the age that you get it.
Okay, interesting. Okay, a topic on everybody's lips at the moment. Is there a possibility for AI to be used in this field? Is it being used, or yes?
Yes, definitely. I am not a AI expert, but yeah, using things like machine learning to take these big data sets and really understand how different individuals are clustering based on a whole host of huge data sets, then you can use that to essentially, essentially once we've got all our big data sets, we can put it into the little black box and work out how individuals are clustering. So we hope to use it moving forward. It won't be me doing that. But a lot of other groups within the space of complex disease, not just Parkinson's disease, are using IA for a whole host of different things, including things like even trying to recapitulate gene editing and gene knockouts using these AI models as opposed to actually having to do the lab experiments. So one day I'll be made redundant, but
somebody has to manage it, surely.
But, yeah, exactly. And you need the experimental data in the first place to actually feed into these machine learning and AI models. But yes, it definitely has a space and in this field and can speed up a lot of things that we do.
Yeah, exciting, very exciting.
Yeah.
Another question from Rich. There is also the longitudinal Parkinson's research being led from the the NZ Brain Research Institute in Christchurch, which might be of interest for those wanting to be involved. So I think that could be for anyone wanting to be involved in research on Parkinson's, the New Zealand Brain Research Institute. But again, feel free to get in touch with us at the foundation. We can try and link you with somewhere.
Yeah, that's at the New Zealand Brain Research Institute is where we got the original cohort for the GBA work. So yeah, links into that.
Fantastic. Yes, it's all connected, isn't it?
Yeah.
Okay, there's one more question. So if anybody does have anything more, please send it on through. But when you were talking about, I think, trauma or brain injury leading to Parkinson's, can you tell at the time of the injury, or for somebody with brain damage, how do you know if Parkinson's applies? Is that written in the genes, or is it more impactful because of what's happened in an accident?
Yeah, so I think if we again go back to the mountain model, the idea was that your genetics kind of set the scene, and then depending on your different environmental exposures, you might go on to develop Parkinson's disease, or you might never develop, even if you've got a fairly strong genetic risk profile to develop Parkinson's. So you could have a huge amount of head trauma and have a genetic risk profile, which essentially means you won't ever get Parkinson's. And because of that interaction, you're probably still at a low risk. But if you've got the head trauma plus a fairly high genetic risk for Parkinson's disease, then you could be more predisposed. But there's no direct links yet, and it's still in its early stages. I think it's mainly most of the research being done is looking into sports players such as NRL and rugby and boxing and things like that. So individuals with repeated trauma, with a main focus, I guess, to see if there's things that can be done to prevent the head trauma happening in the first place, or if there's protective mechanisms that can be used. But yeah, that's still a very early field. But there's down at the Auckland Bioengineering Institute, they're doing quite a lot of research into actually modeling the effects of head trauma. So I think watch this space, not from me, but from other people in that area. But yes.
Yes, wow, that's so very interesting. And as you say, these things can take a long time, and so if you're just starting it, it can take decades sometimes.
Yeah, and it's only a field of research that's been sort of identified very recently.
Absolutely, watch the space, as you say. We have had one more question come through, so we can squeeze this in. How can your research be developed further? I love this. How can we keep this going? Are there any other diseases that are symptomatically or functionally similar to Parkinson's that could benefit from these findings? Or is that not really how this disease research works? Just curious.
Yeah. I think there are quite a lot of diseases. Well, obviously, there's a lot of complex diseases that are similar to Parkinson's disease. And this approach can obviously be taken to a lot. The reason we're interested so specifically in Parkinson's is because of this huge heterogeneous nature, whereas a lot of other diseases are more well-defined. But I think, yeah, things like Alzheimer's and Parkinson's and MSA and different things where the lines are sometimes a bit blurred, you could potentially try and take a cohort of all these individuals and actually see what's similar and what's different from a genetic perspective. But yeah, I think time's going to run out very shortly.
Yes.
Talk about more if you wanted to email.
Yeah, no worries. And again, for anyone who is wanting to get in touch or get more information, please do get in touch with us at the foundation. We can link you in with Sophie or anyone else. But that is the end of our session. So thank you very much, Sophie. That was such an interesting talk, and it was really exciting to hear about such amazing work that you're doing and how it's pulling in all these other teams, even overseas. It's really, really exciting. So thank you.
And thank you. And thanks for the funding too. It's really helps get us to do the research that we want to do.
Yeah, absolutely. And yes, thanks to everyone at home who helps to contribute to that with support. As I mentioned earlier, Sophie was in our Headlines magazine, which came out a couple of weeks ago. For anyone wanting to read that, you can see it online on our website now. It's right there on the homepage. Just click on through and have a bit of a read about Sophie and some other Parkinson's research that we are highlighting. Next month, we will be back online with Dr. Christina Buchanan, who will be talking about Parkinson's and the links with the Pacifica gene. So thanks very much again, everyone, and we look forward to seeing you again soon.
Thank you.
Ka kite.
Bye.